Hello, everyone. My name is Dalila Destanović, and I'm really honored to have the opportunity to present our research at first DNA Aqua International Conference. My team and I have done some research regarding DNA barcoding of Trichoptera in Bosnia and Herzegovina. Before we start, I would just like to go over an overview and as you can see here, this presentation is comprised of 4 sections, and each section aims to answer a particular question. First, I would like you to pay your attention to this hydrological map of Bosnia and Herzegovina. And here you can see that we actually have an impressive river network. We also have a lot of mountains, various microclimates, and when you take that into account, you can see why Bosnia and Herzegovina is generally considered to be very rich in freshwater biodiversity. But what is the status of DNA barcoding in this country, especially when it comes to caddisflies? To answer this question, we have screened the bulk database and we have found almost 59,000 records from 92 countries submitted from them, and not a single one was submitted from Bosnia and Herzegovina. The only attempt that was done by a domestic institution was in 2018 to research DNA barcoding of Trichoptera in this country, and it was made by some of my co-authors. They have managed to show the application of DNA barcoding in pairing of adult and larval stages. They did this on an endemic species, Reacophilida bosnica, and through this process they have generated the first DNA barcode for this species. However, it was never submitted to the bulk database. It remained only in the NCBI. Although this was a great first step to begin researching this area in this country, however, we still had a lot of work to do. So we had to figure out our next step. And a good next step was to investigate 5 protected areas of the Sarajevo canton. And on this slide, you can see those 5 protected areas. We have done our field research according to the ACAM methodology. Here you can see some more detail about our research process. We have collected samples of larvae. We have collected 684 specimens. Also, we have done physical and chemical measurement of each sampling site. And once we got back to our laboratory, we have done morphological analysis of larvae. And we have done this through Atlas of Central European larvae of Trichoptera, written by Waringer and Graf. And we have selected a subset of our samples based on the preliminary morphological identification for further genetic analysis. We have selected 37 samples in total. This genetic analysis included DNA isolation, electrophoresis, and amplification of cytochrome oxidase subunit 1 using JJ primers. Then we have sent those PCR products for sequencing by outsourcing. And once we got those results back, we did some bioinformatic analysis. So, what have we found in this sample? We found that our specimens actually belong to 6 families, that we had 11 genera and 14 taxa in a sample of 37. And in this sample, we have found 24 haplotypes that belong to those 14 taxa. And out of those 24 haplotypes, 20 were not present in bold. On this slide, you can see a table where you can actually compare results of our preliminary morphological identification and identification by DNA barcoding. In this table, you can also see that we haven't managed to identify every single specimen to species level. Actually, 46% of our sample remained unidentified to species level. And also, I would like you to pay attention to Stenophylax equax, Stenophylax nycterobius, and Hydropteryx sp, because these 14 samples actually had the same preliminary morphological identification. And we have thought that maybe we have 1 or 2 species in this sample. However, we actually had 2 genera. And because it would be too difficult, well, speaking of time that I have here, to present all 14 taxa here, I have selected a few ones that would actually show why our research is meaningful. First, I will talk about family Limnophilida because it was the most common in our sample. And genus Stenophylax and Hydropteryx actually belong to this family. So, genus Stenophylax had 2 species in our sample. 6 specimens actually belonged to Stenophylax nycterobius, and they all had the same haplotype. However, this haplotype was not known in the BAL database. We had match of 99.69%, so it was a new haplotype for this database. Regarding Stenophylax equax, we actually had 2 haplotypes in 2 samples, but both of them were previously known in the BAL database, so we had a 100% match. Also, in this overview tree, you can clearly see that we have found 2 species. They clearly separate. Regarding genus Hydropteryx, we have had 7 sequences, and these 7 sequences separate into 3 clusters. And when you calculate their distances, and when you actually run these sequences together with other sequences from this genus, you can see that they clearly separate in 3 different clusters, which means that we likely have 3 different species. However, because these sequences aren't similar to any of those that can be found in the BAL database, we can't guarantee that we have 3 species, and we can't know which species are these. And on this phylogenetic tree, as I said, well, overview tree to be more correct, we have 3 clear clusters. Also, another interesting finding was in family Reacophilida. We had 10 samples from this family. 9 were from species Reacophila fasciata. One remained unidentified. And in this sample of 9 specimens, we actually found 5 haplotypes, and not a single one was previously known in the BAL database. These sequences that we have generated were used by this group of scientists to describe a new subspecies, which proves once again that biodiversity of Bosnia and Herzegovina is far from fully uncovered. To sum up, I would just like to discuss problems that we have encountered through our research. So, sequences that we have obtained were missing from the database, so we couldn't identify 46% of our sample to species level. This is contrary to the general notion among scientists that European Trichoptera are very well covered in the BAL database. Also, another problem that we had was that morphological data on larvae was largely missing. We used a key that is suitable for Central European region. We don't have a key for Balkan Peninsula. Also, something that is always a problem here, it's insufficient financial support. And that's the main reason why we could only analyze 37 samples. Bosnia and Herzegovina does not recognize DNA barcoding as a valid method. They don't have a budget for research or implementation of this method in any field. So it's safe to say that our country lacks systematic approach when it comes to DNA barcoding. As an ending note, Bosnia and Herzegovina could greatly benefit from DNA barcoding. If we used it, we could finally comprehend our biodiversity. And once we comprehend what we have, we can make proper action to conserve it. And also, Bosnia and Herzegovina could greatly benefit from DNA barcoding in freshwater bioassessment. And I'm really thankful for your attention. It's Q&A time. Thank you.